Hi friends, welcome back to my channel Only Bio, presenting up the revision of the chapter Biotechnology. Friends, these are the revision points from biotechnology, and this is the second part of this chapter. Hope you have started following the series. I have already uploaded the first part and it's been uploaded in both the versions, English as well as in Hindi. So all those who are from the vernacular medium and uh, who have a problem in understanding English, so kindly please switch on to that video and please watch that video. The neat points, yeah, the important points for the competitive exams and the MCQs for the same, I shall be uploading it very soon, as soon as possible, or as soon as we end this chapter. So please keep a check on the playlist as well. So we begin with the classical or the elementary concept of the gene. Johansen coined the term gene, and from that time onwards, the term factor was replaced by gene. I hope you remember that the term factor was the term given by Gregor Johann Mendel, who had called it as factor or particle or determiner. According to classical concept, gene can be defined as a unit of chromosome and hereditary material which is responsible for the expression of a character. Friends, we all know that chromosomes carry the genes, or rather chromosomes are the carriers of genes, and genes are DNA molecules which basically act as a hereditary material and are responsible for the expression of a character. The important aspects of this classical concept of gene are: first, the genes are hereditary units which are transmitted from one generation, that is parents, to the next generation, that is offsprings. Second, during this transfer, the genes are present on the chromosomes of the gametic nucleus. Friends, gametes carry the nucleus which fuse to form a zygote, and this is how the zygote gets the genes from both the parents. Point number three, the genes are arranged in the linear order on the chromosomes. Linear order is nothing but one after the other. Next, the single chromosome shows presence of many genes. Friends, there is no hard and fast rule that every chromosome must have only X number of genes. It may differ from one chromosome to another. Every gene occupies a particular position which is termed as locus. Friends, chromosomes are the carriers of genes, right? And the point, the place where a particular gene is present is said to be a locus. So plural can be called as loci. Next, alleles or allelomorphs are the alternative forms of the same gene. The alleles may be dominant or recessive depending upon their phenotypic expression. Now, alleles are the alternative forms of the same gene, that is, they are different forms of the same gene. And if the allele tends to express itself, then it is called as a dominant allele. And if it does not express itself, so generally it will be called as a recessive allele. And this all depends upon their phenotypic, that is, external expression. Seventh, the genes existing in more than two allelomorphs are referred to as multiple alleles. Friends, normally we hear of two alleles of a particular gene, but at times there can be many. If you're thinking of how come multiple alleles exist, so please remember this is possible due to mutations and the multiple forms of the same gene. Okay, they are called as multiple alleles. Point number 8. The gene can undergo mutation, and such mutated gene is always recessive in nature as compared to a normal gene. So normal gene is the wild type of gene, the natural gene which tends to be dominating the mutant gene. Point number 9. Gene can be transferred from one chromosome to another homologous chromosomes by crossing over and non-homologous chromosomes by translocation. Friends, when the genes get transferred, then and only then there are chances of variation. Normally, the genes can be transferred only during the process of crossing over that occurs during meiosis. If you remember, it occurs during Paxton stage of meiosis 1. And if the chromosomes are non-homologous, then the only possibility of the genes to be transferred on the non-homologous chromosomes can be the process of translocation. In other words, crossing over occurs only between the non-sister chromatids of the homologous chromosomes. Friends, if you are trying to recollect what are homologous chromosomes, so let me remind you, the chromosomes which have a definite size, shape, structure, they are identical with respect to the genes they carry, are termed as homologous chromosomes. Let us understand the modern concept of gene. It is the result of work of different workers in the field of genetics, biochemistry, and molecular biology. It was formulated in the first half of 20th century. In 1928, Frederick Griffith, scientist working on disease-causing property, that is pathogenicity of a bacterial strain Diplococcus pneumoniae, discovered the process of transformation in which he carried out experiments on pathogenic and non-pathogenic strains of Diplococcus pneumoniae. Friends, let me remind you that pneumonia is a disease that's caused by a bacterium called as Diplococcus pneumoniae. And this bacterium has two strains or types in it. One that is pathogenic and the other that is non-pathogenic. So this was being found out by Frederick Griffith because he was conducting his experiments on the property of pathogenicity of Diplococcus pneumoniae. Pathogenicity is nothing but it's the ability to cause disease. Later on, experiment was repeated and was explained properly by Avery, MacLeod and McCarthy in 1944. They concluded that the conversion of non-pathogenic strain into pathogenic strain was due to transformation of nucleic acid, that is DNA, and not due to proteins. Friends, there was an experiment which was being conducted. I think you might be recollecting it. So, it was observed that the non-pathogenic Diplococcus pneumoniae, which were not supposed to bring about pneumonia disease, still it was found that they become pathogenic and bring about that disease. So scientists were slightly confused with respect to this, and they were confused with respect to this in what sense? That is, it could be either the bacterial DNA which is bringing about this conversion or the proteinic part which may be bringing about this conversion. But later on, by the experiments done by these all scientists, it was concluded that it can't be proteins which change the non-pathogenic to the pathogenic strain, but it has to be only DNA. Thus, they proved that genetic material transmitted from one generation to the next is the nucleic acid, that is DNA. Likewise, Lederberg's experiments conducted in 1948 of transduction with bacteria and T-even bacteriophage, that is virus, confirmed the nature of genetic material as nucleic acid DNA. This was further confirmed by Hershey and Chase in 1952, that is DNA is the sole carrier of genetic information from parents to offsprings. Friends, remember bacteriophages are the viruses that infect bacteria. So it was being concluded, confirmed that it is only and only DNA which acts as the genetic information, or it carries it from parents to the offspring. So now it is known that gene is made up of DNA and consists of nucleotides. Friends, hope you like it. Please subscribe if you haven't yet done it, and also hit the bell icon so that you get to know my new videos. Thank you so much for being with me.